Okay, good morning, everyone. We're going to get started in the guise of timeliness. Just a couple of things, as always. Thank you so much for joining us to our weekly research bite in place of our annual conference. We will be muting all of you, but if you could also do us a favor and mute yourself until the end of Natalie's talk, that would be great, just so that we don't end up having too many distractions during the presentation. And then during the question period, it'll depend really on how many questions come through the chat, and we're going to ask that you use the chat function rather than the question and answer function if you have any questions for Natalie. If there seems to be some really great questions that would engender a bit of a discussion, we'll go that way. Otherwise, you can just fire your questions either at Andrea or I, and we will then moderate the discussion afterwards. So it is my great pleasure to introduce Natalie Bell, who is currently a second-year master's student in the Department of Laboratory Medicine and Pathobiology at the University of Toronto. And those of you who've been around INCI for a while know Dr. Samira Mubaraka, who is one of the big foremost leaders on the cusp of COVID research right now. So that's the lab that Natalie is actually working in. So we're really honored to have her here as she discusses some of her research that includes large field sampling, investigating the use of environmental sampling at the farm level for avian influenza and swine. So it's over to you, Natalie.
Thank you. Thanks for the introduction. So my project is environmental sampling and next generation sequencing as a novel approach for the detection and characterization of Influenza A virus in swine. Next slide. So the Influenza A virus is part of the Orthomyxoviridae family, and I'll be focusing primarily on Influenza A virus, which is a segmented negative sense RNA virus. And there are 8 genomic segments. 2 of these segments, which code for the surface glycoproteins. So this is the neuraminidase and hemagglutinin glycoprotein. So there are 18 known hemagglutinin and 11 known neuraminidase. Next slide. And so the hemagglutinin receptor, or the hemagglutinin glycoprotein on the outside of the virion binds to the host cell receptor, which is a sialic acid. And depending on the host of the virus will actually display certain specificity towards the sialic acid linkage. So mammalian influenza tends to bind the alpha-2,6 linkage, whereas the avian influenza tends to bind the alpha-2,3 linkage. But this isn't a hard and fast rule, and there are some mutations in the receptor binding domain that can actually change this affinity. Next slide. And so one of the greatest advantages of the Influenza A virus is its ability to evolve, mainly through antigenic drift and shift. Recombination also occurs, although to a lesser extent. So antigenic drift are these small changes that occur over time. However, the antigenicity of the virus typically still remains the same. Whereas with antigenic shift, this is a major immunological change. And so this occurs from reassortment. Next slide. And so, as I mentioned before, it's a segmented virus. And so if a host cell is infected with 2 or more different influenza strains, when the viral progeny are being created, essentially they can have segments from the different parent influenza viruses that have affected that host cell. And so one example where this occurred was with the triple reassortment H3N2 influenza virus, and this emerged in swine populations in North America in the late 1990s. And so, as I mentioned before, again, with the receptor linkages, so swine actually contain sialic acid receptors with both the alpha-2,6 and alpha-2,3 linkage. So essentially, they can serve as a mixing vessel for reassortment events like this to occur. Next slide. And so why is it important to have surveillance for swine? So influenza A isn't reportable under the Canadian Federal Animal Health Act. There are concerns for pandemics. So for example, the H1N1 pandemic in 2009, which originated from swine. And Canada is actually the 7th largest pork producer in the world. So since there's a great frequency of trade, including live pig exports and imports, these can also serve as occurrences for reassortment events. Next slide. And so our lab has a bit large focus on bioaerosol sampling. And so bioaerosols are these fine particles that are derived from a biological source. They can range in size and composition, and they're suspended in the air. So typically, bioaerosol sampling is a non-invasive technique that's used primarily for non-viral microbial agents, so mainly bacterial and fungi. And the data related to the detection of airborne viruses is a lot more limited. However, there have been some studies that have detected airborne virus, specifically in swine barns, which are listed below. Next slide. So for this study, we collected samples at a farrow-to-feeder swine facility in southern Ontario, and the number there, 16, represents the amount of times that we visited the barn. And we also had a negative control run here at Sunnybrook Research Institute. They have pigs here that are housed for imaging. And so the 3 sample types that we collected are oral fluids, air samples, as well as environmental swabs. Next slide. So to collect the oral fluids, we attached a cotton rope into each of the pig pens. And so in each pig pen, we had around 30 pigs, and there were 8 pens per room. And so we collected 2 pooled oral fluid samples per room. And the oral fluids are what we used as a positive control. So the oral fluids basically told us if the room was positive or negative for influenza A virus. We also collected environmental swabs, where we took a flock swab and we swabbed a feeding tray. Next slide. So for our bioaerosol collection, we used 4 different collection devices. So the 2 on the left, so we have a PTFE filter and a NIOSH 2-stage cyclone sampler. And so these are our low volume samplers, where we sampled at 3 liters per minute. And then the 2 samplers on the right, so the Coriolis and the SAS dry air sampler, are the large volume samplers, where we sampled at 300 liters per minute. Next slide. So I'm just going to briefly go over threshold cycle, or CT value, since a lot of the output for this project was using qPCR. And so the CT value essentially is the cycle number where the fluorescent signal crosses the threshold, and it's inversely related to the amount of your target. So the lower the CT value, the more of your target. So in our case, influenza A virus in our sample. Next slide. So these are the results from the barn. So along the left-hand side, you can see the types of samples that we collected. And along the top, you can see positive, suspect, or negative samples. So in the positive column, you can see that 87% of our samples of the oral fluids were positive, and 43.8% of our environmental samples were positive by PCR. And then I also want to look at the suspect positive column. So what you can see is that the air samples actually had a lot more suspect positives compared to positives. And this was based on the CT value of 36 on our qPCR. Next slide. So here we're looking at the positivity by sample type with a positive oral fluid. So as I mentioned before, the oral fluids are a positive control to determine if the room was positive. So along the x-axis, you have the different air samplers as well as the swabs. So what you can see here, so the black bar is the positive samples, and the gray bar is the suspect positive. So again, that's based on the CT value of 36 as our cutoff. And for the air samplers, we have a lot more suspect positives than we do positives. And so even in rooms where we knew that there was influenza A virus, we weren't always able to pick it up in the air or in the environmental swabs. Next slide. I also wanted to look and see if the air samplers, if there was any difference between the amount of copies per liter of air of the influenza A virus RNA that we could find in the air. However, there was no significant difference between the air samplers. Next slide. So once we knew that we can detect influenza A virus in our air samples as well as the environmental swabs, we then wanted to see if we could derive viral RNA sequence from these samples. And so we use amplicon-based sequencing. And so the sequencing was actually done at National Center for Foreign Animal Disease. Next slide. And so I'm not going to go over the bioinformatics pipeline that we use to analyze the samples in great detail. I'll just go over it briefly. But after these samples were sequenced, we then used spades for our de novo genome assembly. We then blasted our samples against the Influenza Research Database and perform a reference-based alignment with bowtie 2. To assign operational taxonomic units, we use Influenza Research Database, and we also assign taxonomy using CHIME. Next slide. And so here on this graph, along the y-axis, you're looking at the coverage of the influenza genome as a %, and along the x-axis, you're looking at the samples based on their CT value. So the oral fluids typically had lower CTs in the 20s range, whereas the environmental samples, so the air and swabs, typically had CT values in the last group, so the higher CT values. And what you can see for this group is that the coverage of the genome is a lot more variable. And again, this is what we see with the air samples and the environmental swabs. Next slide. I also wanted to look at the breadth of coverage by segment as well as sample. So the blue bars are the oral fluids, the green bars are the environmental swabs, and the pink or red bars are the air samples. And the coverage between the oral fluids and the air samples, so the air samples were significantly lower in coverage for almost all of the segments. However, you don't really see a statistically significant difference between the environmental samples and the oral fluids. So this is showing that the environmental swabs may actually be just as good as the oral fluids in terms of coverage. Next slide. And so this is just a mini phylogenetic tree of the different strains of influenza A virus that we detected through sequencing. And the different colors represent the years of the influenza A virus that it was first detected. And so just to reiterate, these are samples that were collected all on one farm. So even on one farm, we could find 13 different influenza A virus strains. And this, again, is evidence for potential reassortment events to occur between the different strains. Next slide. So we also wanted to look at alpha diversity by sample type. So this is the diversity within. So along the y-axis, we have the Shannon index as our alpha diversity metric. And along the x-axis, there are the different groups of sample types, so oral fluids, air, and swabs. We didn't see a significant difference between the diversity within each sample type, though. Next slide. And lastly, we wanted to look at how the environmental samples and the oral fluids compared in terms of clustering. So the red circles are the environmental samples, and the blue squares are the oral fluids. And so this multivariate analysis here is just showing that the environmental samples as well as the oral fluids, they cluster differently, and this is based on an OTU clustering, so on the operational taxonomic unit. And we defined in OTU as a specific segment from a different strain of influenza A virus. And you can see that there's a difference between the OTUs that were picked up between the environmental samples as well as the oral fluids. Next slide. So in terms of future direction, something that we're working on right now is we're looking to identify phenotypically characterized SNPs, as well as mutations that confer increased virulence, as well as mutations that either increase resistance or decrease susceptibility to antivirals. And so that's something that we're currently working on. Next slide. So in summary, we did see that we can detect influenza A virus in our environmental samples as well as derived viral RNA sequence, and that the co-circulation of the different influenza A viruses demonstrate a potential for viral reassortment. The increase in CT value, which was seen in the environmental samples, which shows a lower % coverage of the influenza A virus genome. And you can see that the breadth of coverage for the air samples are significantly lower than the oral fluids. So although air samples may not be able to be used as although air samples may not be a standalone method for surveillance, they may be used as a complementary method to environmental surface swabs as well. Next slide. So I'd like to thank my supervisor, Dr. Samira Mubareka, as well as my committee members for their advice on this project, as well as the members in my lab, especially Dr. Hamza Marash. He's a postdoc in our lab, and without his help, I wouldn't have been able to do the bioinformatics analyses, as well as Dr. Karen Prost, who had started this project before I came in. I'd also like to thank the members at NCFAD who were able to perform the sequencing for us on our samples. Next slide. So thank you for listening. And if you guys have any questions, I can take them now.
Thanks, Natalie. That was phenomenal. And I'm sure that it took an awful lot of, a lot more than the 2 years that you've been in there to come up with all of this. That looks quite in depth. So if you were going to go on and do a PhD, would you be interested in pursuing further work on this, or what are your plans?
Sorry, what are the plans for the project?
No, for you.
I'm not sure. I'm actually pretty interested in policy work and public health related work, so I'm not sure that I want to pursue a PhD in basic science, but maybe something public health related. But in terms of this project, I think there's still more analysis that can be done. I think there's a lot more analysis with the SNP that we're looking into now. And even just in terms of bioaerosol and environmental sampling, there's still a lot more that I think needs to be done in the field.
Right. And one of the reasons I was asking is because we know that there's a propensity for individuals to end up working in a silo, right? And this is something that looks like it would be very important in the field of public health, where it would be, such as with COVID, good for multidisciplinary work to happen. So there's a question just with regards to the different viral strains found in oral fluids in the environmental swabs. Can you expand a little bit on that?
Yeah, so I guess it's based on the way that we defined our OTU. So that was the way that we analyzed our samples. So we define an OTU as a segment from a specific strain of Influenza A virus. And so in terms of the differences between the environmental swabs and the oral fluids, we tended to find different strains between the two. We actually saw there were two strains that came up quite frequently. There was a Manitoba strain and an Ontario strain that came up in a lot of our samples as well.
Okay, and so the diversity of the strains across geography, how closely related are they? Like, so not being a virologist, how different would they be? And is that something that needs to be watched more closely?
So in swine, the endemic subtypes are H3 and H1. So the hemagglutinin proteins and the neuraminidase proteins are mainly where you can tell the differences. A lot of the other segments are pretty conserved. So, for example, like the matrix segment is conserved amongst the different strains. So the main differences, and what we're looking for especially for SNPs, are the hemagglutinin and neuraminidase. But we didn't actually pick up any other subtypes that would be out of the ordinary. So we mainly just found the H3 and the H1 subtype, which again are endemic in swine.
That's good to know. We aren't developing yet another pandemic. Does anybody have any other questions? Feel free to unmute yourself, which is *6. Can you comment on the practicality of widespread use of air sampling for surveillance in swine farms?
So it's currently not being used for airborne detection of viruses. It's something I guess still relatively new, but due to its feasibility, I think that ideally you'd want to have these bioaerosol samplers set up within swine barns. But I think at this point right now, I think the coverage is, in terms of sequencing, the coverage is still fairly low. So I think there needs to be more optimization in the methods in order to actually recover more viral RNA. But yeah, due to the fact that it's non-invasive, I think that more research needs to go into it in terms of how we can actually use it in the field, because it is quite easy to use, but I just think that in terms of the results and coverage, it's not quite there yet.
Okay. And was WGS used for sequencing?
Yeah.
And for those people on the call who don't know what that is, would you like to actually explain that?
Yeah, so the sequencing was done on a MySeq Illumina-based platform. So I actually didn't do the sequencing. The sequencing was done over at NCFAD, but we used amplicon-based sequencing, and it was whole genome sequencing.
Okay, great. And with respect to the differences between environmental and oral fluids, you need to take both types of samples in order to detect influenza. So if we were actually going to be looking into an outbreak, for example, what do you recommend as the best way to actually ensure that we get adequate samples that are actually predictive of what it is that we're seeing?
So, I mean, the actual gold standard would be to use an NP swab in a pig, but that's a pretty invasive method. So there was a paper by Susan Detmer who basically verified in oral fluids as basically a more non-invasive approach to actually pull influenza A virus in swine. So to get influenza A virus from environmental swabs and air samples is more new and maybe not as verified. So the oral fluids were our positive control to really determine if it was there. And we want to see if we could get some similar results in the air and swabs. But ideally, the actual gold standard would be to swab the nose of the pig.
Okay, great. Does anyone have any further questions? So, Natalie, how closely related would you say what you're seeing now is to what was seen in the previous swine flu epidemic? So how many minutes? I'm not sure I understand that question.
Sorry, I missed the question.
Oh, so the question is how closely related would you say the viruses that you've identified currently are to the historical swine flu that created the global epidemic?
We haven't found any evidence of the pandemic strain within this swine barn.
Okay. And so we have a whole bunch of people saying thank you. That was a great talk. Does anybody have any further questions? Feel free at this point in time to unmute yourself, and that's *6. And so far, none. So what we'll do, Natalie, thank you. You were actually the most prompt speaker we've had so far, which is partly why I think everybody's running out of questions, is you actually finished a little bit earlier. So good job. You didn't run on at the mouth like the rest of us did. But thank you so much for sharing your research. I'm sure there will be questions that end up being sent to me that I'll then forward to you and then forward out to everyone else, the responses. So thank you, everybody, so much for joining us this week. Thank you to Andrea for running the slides as well as she always does. And next week, we will have Dr. Ryan Brooks, who will be talking about some of the issues surrounding modeling and mitigating the issues of wild pigs in Canada. So have a great week. stay safe and wash his hands.
Thank you.